A 1-hour primer to mitochondrial genomics. And my background is a computational biologist. I've been working with Vamsi Mutha, who's a PI studying mitochondria here. I've been working with him for 10 years on this. And feel free to interrupt me, ask questions. This is for you guys, so let's get started. Maybe. Oh, the Google Calendar says it's time to give a talk. All right, try this again. All right. So let's start with why you should care about mitochondrial genomics. So right here, while you're eating your breakfast, you're not doing too much work, but it still takes about as much power as running a 100-watt light bulb to provide the energy for all of your metabolism in your body. Now, about 20% of that is going to your brain, even when you're not thinking that hard. And mitochondria generate approximately 90% of the cellular ATP that's used in all of metabolism. So in order to do our body's work, there are two main sources of generating ATP. One is glycolysis, and the other is a membrane-bound system of oxidative phosphorylation. Who of you have learned this, who have not learned this in biochemistry class? Is this new to you? A couple news. Okay. So this is one of the coolest processes in all of evolution. This evolved pretty early on. It's present in almost every species on the planet. A couple have lost this ability. And it uses a membrane and a gradient across a membrane to drive a machine that turns ADP into ATP. It sticks an extra phosphate and turns adenosine diphosphate into adenosine triphosphate, and that is the supercharged molecule that then is used, it's like a battery, it's used to power almost all of the metabolic reactions in the cell. So how does this work? So here's the mitochondria here. All these little things here are the inner membrane, shown here as these blue lines. So there's 5 complexes that are just jam-packed on this membrane. And the job of the first 4 of them is to pump hydrogen to the outside of that membrane. And this creates an enormous electrochemical gradient across the membrane. So it's electric because there's positive charge on one side and negative on the other, and it's chemical because there's the hydrogen excess on one side. So that huge electrochemical gradient drives this ATP synthase, also called complex 5, and it looks like a water wheel. It's a motor. It's a set of proteins that act as a motor, and it's like, imagine a dam with water flowing over the dam, and it literally turns this motor, and every time the motor turns, it jams a phosphate onto an ADP and creates an ATP. So that's the phos in the oxphos. That's the phosphorylation, and that's what creates ATP. The ox part is how do you actually pump protons against this gradient without using more energy than you're getting at the other end. And this is the real challenge. And the solution that evolution has come up with is amazing. It all starts with high energy electrons. So all the food that you're eating right now gets converted into smaller parts, into acetyl-CoA, and that's fed into the Krebs cycle, which it's like a crike. It turns these small molecules into even little smaller bits, including high-energy electrons, and then waste products like carbon dioxide. Those high-energy electrons go here to complex 1 or complex 2, and you have this high-energy electron, and then it bounces down energy states, and each time it goes down an energy state, it pumps, or it moves the machine and lets one proton go from one side of the membrane to the other side. And these are very complicated machines. Complex 1, the crystal structure has recently been solved. It works like a piston. So in this part here, the high energy electrons are coming in, they're jumping from iron sulfur cluster to iron sulfur cluster, and that causes a conformational change. It works like a piston. It opens up these tiny, tiny little columns of water, and then a proton on this side ends up on the proton on the other side. And that's how complex 1 works, similarly 3 and 4. So these electrons are starting at a high energy state, they're going down, 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 and at the very bottom, they have to land somewhere that really wants that electron more than anything else. And that is oxygen. So this is free oxygen. This is the main reason you have to breathe is the oxygen used to suck up these electrons and drive this process of high energy electrons to low energy electrons. And it takes oxygen and creates water. It's a great waste product. Water, it's a lot of, so there's not a lot of toxicity with this product, but you need oxygen. If you don't have oxygen, you can't, the high energy electrons aren't going to want to go to the next phase, So if there's a lack of oxygen, the whole system breaks down. If one of these complexes is jammed, the whole system breaks down. So you need these four complexes to work. They create this proton gradient, and then that is dissipated in this ATP synthase, generates 90% of our cellular ATP. So are you now interested in mitochondrial genomics? It's really cool. Okay, so we breathe oxygen all the time. 90% of the oxygen that we use in our body is used in this reaction. There are other reactions that use oxygen, like in the peroxisome and some other places, but this is really the main one. So how much ATP is this generating? Well, at rest, we have about as much ATP as would fit in a teacup in our body, but it's constantly being turned over all the time. So if you wanted to fill your body's ATP needs by ordering it from Sigma and eating the ATP, you would need to eat 140 pounds of ATP every day. That's how much we are constantly being generated. So it's continually going in a pool between ADP and ATP, but I'm trying to give you a sense of how much ATP that mitochondria is generating. So in order to do that, you need a lot of membrane. So these structures are designed to have a lot of surface area. They're jam-packed with these membranes with very little space on this side. So it's a double membrane in the mitochondria. This has a very little space so that you can create this enormous gradient over a small space. 120 millivolts is created here. So if you took all of these mitochondrial membranes and stretched them out, it would be the equivalent of 2 soccer fields. Is jam-packed into this real estate in our mitochondria in our bodies. And this is the real system by which all of eukaryotic life is powered. There are a couple exceptions of organisms that are anaerobic, so they have lost this whole system. They get their ATP from the organisms that they live near. They're usually parasites. But pretty much every other eukaryotic organism is powered by mitochondria. So that's just the background of why you should care about mitochondria. Now I'll go into the genomics. I'll give you a history of mitochondria and then talk a lot about the human genome, of the human mitochondrial genome, then a little bit into how this evolved in eukaryotic evolution, some of the technical challenges of analyzing the mitochondrial genome because it's different from the nuclear genome, a big chunk on mitochondrial disease and how we diagnose it. And if there's time, end with somatic mitochondrial changes in tissues and in cancer. Any questions before we get started? Okay, so mitochondria. The name comes from the word mitos, because they look thread-like, a Greek word, or chondrion, which looks like a granule. So very early on, people realized that these both looks like threads and granules. And these were discovered by beautiful studies in the 1840s. The initial founder called them bioblasts, and they were renamed in 1898 to mitochondrion because of these two states. Now, you probably are used to seeing them in this state, which is often an artifact of EM, but in reality, mostly they form a reticular network, kind of like the ER. They're constantly fusing and having fission. They're moving around the cell in this state here. The yellow and the red is the mitochondria, with the blue is the nucleus. Now, here are some EM images of mitochondria from different tissues. Now, the main function of mitochondria is this energy production system, but it also is the center of a lot of other biological pathways that reside in the mitochondria. So depending on what the cell type is doing, the mitochondria look structurally different. From heart, which have these beautifully stacked cristae, because the heart's really trying to generate ATP, to other tissues that are doing more biosynthetic processes. And like the liver, it's doing a lot of steroidogenesis. There's less ATP role and more room for other things that are happening in the cell. The cristae here, the word for these mitochondrial membranes inside are called cristae, and that's this energetically charged part. And they actually can, even though it's this network, there are little power relay stations between mitochondria that are touching. This has been recently appreciated just in the last couple of years, and there's thought that these act like power relay stations. So if you're trying to pass, to create this charged mitochondrial network in a muscle cell, the part that's using oxygen is sitting, the 4 complexes that use oxygen are sitting right near the blood vessels. The ATP synthase is sitting farther down, closer to where the ATP is needed in the muscle. And then you maintain that membrane potential based on these connected mitochondria with these power relay stations. And this is just another showing of how they actually get lined up in tissues. It's pretty cool. Now, the mitochondria don't just act with each other, they also interact with all the other parts of the cell. Here they are next to the ER, and there's many important crosstalk between the ER and the mitochondria. Also, here's lysosomes, peroxisomes, and mitochondria are tethered because the peroxisomes are taking our very long chain fatty acids that we're eating, and they break it down to medium chains. That's fed into the TCA cycle and used to create energy in the mitochondria. So those organelles are also tethered. Depending on the needs of the cell, there are many different numbers of mitochondria. So typically a cell will have some hundreds to thousands of mitochondrial genomes per nuclear genome. The largest is the human egg, which is also one of the larger cells, but it has 100,000 mitochondrial DNAs per nuclear DNA. And here's a study quantifying a relative amount of mitochondria across different tissues. So you can see tissues that have a huge energy demand, like the heart, have much more mitochondria than things that are doing less work, like the lung. The fetus here is interesting. The fetus, of course, is doing a lot of work, but it's not a very high oxygen environment, so it uses a lot of glycolysis. And so it's not as dependent on the mitochondria as you would think. So as you probably all know, the reason I'm here talking in mitochondrial genomics is that the mitochondria have their own genome. Unlike any other organelle in the cell, here is a picture of it. It's a beautiful circular genome. Here it's replicating, so it has 2 copies. And if you're looking at this, you may be thinking, that looks a lot like bacteria. Well, there's a reason for this. Mitochondria derived from an endosymbiosis of a bacteria into an archaeal host, way at the dawn of eukaryotic evolution over 2 billion years ago. And a lot of the features still hark back to this bacterial ancestry. The human mitochondrial genome is tiny. So it has its own genome, it has lots of interesting features, but size is not one of them. It's 16 kb. To put that in context, a typical gene in the human genome would be about 75 kb. This one is 16 kb. It's tiny, but it's jam-packed with information. There's almost no part of it that is not coding for information. So in these 16 kb, there are 37 genes. 13 of them are protein coding, and they all code for membrane subunits in that OXPHOS system that I showed you earlier. And then there's some tRNAs and rRNAs needed to turn these 13 proteins from RNA into protein. The mitochondria genetic code is similar to the regular one in the nucleus, but there are a couple important differences. Some codons that in general in the nucleus code for arginine now code for stop. There's a different one for isoleucine, and UGA, which is a stop codon in the nucleus, now codes for tryptophan. So you can imagine some differences here. If you have genes that are migrating from the mitochondria to the nucleus, you may have some differences in how they would be coded for. The mutation rate is about 10 times higher in the mitochondrial genome than it is in the nuclear genome. Yes, question?
There are 100,000 copies in the egg.
Yeah.
Does that mean that the mitochondrial genome is more heterogeneous than the somatic nuclear
Absolutely.
genome?
So I'll get to that. So that's a great point. So if you have these self-replicating little mitochondria, they can pick up mutations and they reproduce asexually. So all of the children of one mitochondrial genome will look like that one. There's no sexual reproduction. And so you can end up with, within one cell, like the egg, you can have different mitochondrial genomes with slightly different sequences. Yeah?
Is there an explanation for the slightly different genetic code?
Yes, so in fact there are about 13 different genetic codes known out there. Bacteria have a slightly different one than vertebrates, so I think it's partly a stochastic process. Some of the reasons are quite interesting, which is AGA and AGG, it says it's codes for stop. In fact, there's no tRNA that recognizes these. So they've just lost those tRNAs. And when you have nothing to translate it, it causes stalling and then it stops. So here, the reason that you have this different genetic code is that you lost a couple tRNAs, and now the organism doesn't know how to translate them. So that's one explanation. The higher mutation rate is due to a bunch of different things. There's not as many robust processes for mismatch repair compared to the nucleus. And it is in an environment where there's a lot of ROS, oxidative damage, that can also cause a higher mutation rate. Typically, in the D loop up here, there's areas here that are hypervariable, very different, either in somatic evolution or among people. And this is why when sequencing was hard and expensive, people would just sequence this tiny little bit of the mitochondrial genome to look at ancestry, because it is very different among people. So this is the human mitochondrial genome. I have a star next to 13 protein coding genes because that's what the textbooks would say. There are 2 recent papers saying that hidden in these rRNAs are little tiny peptides that are coding for proteins that are really important. They're a little controversial, so I didn't revise my slide, but it is interesting that stuff is still being discovered here. And the transcription of these genes is much more like bacterial genomes than it is for nuclear genomes. They're polycistronic, meaning that you transcribe almost the whole circular molecule and then it gets broken up, clipped into little bits and translated into proteins, just like a bacteria. There are no introns in animal mitochondria, or at least in ours. So if you think of the DNA is 2 strands. If you move up the temperature, the 2 strands will separate. And if you run them on a gel, one is heavier than the other. So one strand is called the heavy strand. That's because it has more G's and fewer C's. And that strand encodes 28 of the genes. The other strand shown here, the light strand, it only has 9 genes. That's just the way it happened to evolve. And interestingly, there are 2 pairs of overlapping genes. So one little locus actually codes for 2 different proteins depending on where you start. So this is all to show you that the 16 kb molecule is jam-packed with information. Almost nothing, no base here is not being used for some information purpose. One of the most interesting things about mitochondrial genomics is uniparental transmission. So to put this in context, as you all know, we are sexual animals. You need to reproduce, you need 1 set of chromosomes from the mom, 1 set of chromosomes from the dad. The mitochondria, that's in the nucleus. The mitochondria comes only from the mom. And this is not just in animals or in us. This is true across the entire spectrum of eukaryotic life that we know it. Almost all eukaryotes are sexual. There are a couple exceptions, and those tend to be very short-lived and they die out quickly. The mitochondria is always uniparental, and the copy coming from the other cell, in this case the sperm, gets specifically degraded. There is a reason, evolutionarily across billions of years of evolution, to make sure that the nucleus has sexual reproduction and the mitochondria has uniparental. We can go into some of the theories of why, but let me just tell you why we know this. So the paternal DNA, so the sperm coming in, has only like 50 to 100 mitochondria. They're packed in the tail. They let the sperm swim. The egg has 100,000 mitochondria. So first of all, you have a huge dilution factor. Maybe that's why we're not seeing paternal inheritance. Well, you do have dilution, but that's not the full answer. The sperm mitochondrial genomes are specifically imprinted, ubiquinated, and then they get degraded in the egg. And if you block the lysosome so you can't degrade them, then you will see this very small amount of paternal transmission. And this uniparental or maternal transmission has been confirmed through very large pedigree studies. And people have been looking for paternal inheritance and through deep sequencing and PCR to search for paternal alleles, and pretty much there's none. There are a couple exceptions that kind of prove the rule. They did mouse back crosses generation after generation, And they, looking really hard, they could find 0.01% to 0.1% leakage of paternal alleles. There's been a single human case where they see paternally mtDNA molecules that are at 90% in the muscle of the affected child. And that was such an exciting finding that that one case was published in the New England Journal of Medicine. And in a 50-generation chicken pedigree, they found one case. So those are the exceptions, but pretty much there is uniparental maternal transmission of the mitochondrial genome. Yep.
Are there any phenotypic consequences of blocking lysosomal degradation of the maternal mitochondria?
Well, when you block a lysosome, you don't just block this, you block a whole bunch of things. So that's not viable. They just did that to prove it. One of the theories of why you have to have this is if at the beginning I talked about oxphos chain, this is the 5 complexes that is needed in order to create 90% of the ATP energy in our cells. Some of those protein subunits are encoded in the nucleus and some are in the mitochondrial genome. If you have an even slight mismatch between the protein sequence on your nuclear DNA and the one in the mitochondria, that will cause a huge problem, energy deficit, and may lead to death. So you want to make sure that you're going to be able to travel with alleles that are matching. So if you are going maternal transmission, you're always going to match at least half the chromosomes that you're going down to in the next generation.
Another way to get the biology behind that question
is
that there are now several monogenic disorders of ubiquitination
that
have phenotypic consequences. Have they examined whether or not there's any specificity for retention of the male mitochondria in any of those?
That's a great question. I don't know if they've looked for it specifically. I mean, you do have this dilution problem anyway, so you would expect a very small amount to be getting through, but that's a great idea. Yeah.
Are there any RNA or proteins from outside the mitochondria that are part of the working operation of the mitochondria?
I'll get to that, but the mitochondria has about 1,200 proteins that make it up. 13 of them are from the mitochondrial genome, and the other 1,100 and whatever come from the nucleus, and they have to be coordinated in order to work. Okay, so I think this concept already came up before, but in the cell, you have lots of mitochondria. Each one has, you have a lot of mitochondrial genomes. They can be slightly different from each other. And this gives rise to the concept that's called heteroplasmy, which means you have a variant that's present in some mitochondria, say the red ones, and not other ones. And so if you have a deleterious allele, it may be not a problem if you're just at a few copies in the cell, but if you cross some threshold where you're now mucking up most of the copies of mitochondrial genome in the cell, then you can cause a phenotype. And so this is seen all the time in diseases of the mitochondria, where if you have 100% normal, you have no phenotype. Even when you're at 40 or 60% heteroplasmy of some deleterious allele, there's no phenotype. It's only when you cross some threshold that suddenly you're seeing muscle disease or neurodegeneration. So this is a main concept of looking at mitochondrial genomes. One of the interesting things about this that's emerged in the last couple years is that this heteroplasmy, the mutant load, is different across tissues, and it's different across tissues in the same way. So this is a beautiful study of a monozygotic set of twins with a deleterious allele, shown here. So here, the unaffected mom has this deleterious allele at different heteroplasmy levels across different tissue, 5% in the blood, 25% in the epithelium. Then they looked at the monozygotic twins. They had very similar heteroplasmies to each other in the different tissues. And this has now been replicated in another study with another set of monozygotic twins with a different mutation. But this, I think, has two important implications. One is very practical. The blood, in general, if you have a deleterious allele that's causing a problem in the muscle, the blood often just doesn't have it anymore. Blood is very rapidly dividing, and if you have a deleterious allele, those cells may not divide as much, and so they lose out in the population, whereas a terminally differentiated tissue like the muscle can accumulate these very high mutant loads. So if you're doing genetics, in germline genetics, we almost always sample blood because it's really easy. We're going to miss alleles that may be very high in other tissues because we're sampling the wrong tissue. So it's a little bit of a side point, but I think it's important practically. Another slightly side point, but again is practically important, is a concept of nuclear sequences of mitochondrial origins called numites or numps. So what happens in evolution, here in stylized mitochondria with their purple circular genomes, is those genomes can get integrated into the nuclear DNA. And this is happening all the time, both across evolutionary history and just within the population. If in our human genome assembly 19, there are over 700 of these copies of the mitochondrial genome that are now littering our nuclear genome. If you just take the mitochondrial genome, the 16 kb, and blot it against the nuclear genome, you can see all of these different hits. Some of them are really similar, 98.6% identity on chromosome 1, 85% on chromosome 17. So this is the genome, nuclear genome is littered with these ancient relics. And they're actually non-randomly inserted. So there's hot spots where they tend to get inserted. For sequence analysis, it's not usually much of a problem because the nuclear genome and this numite might have 2 copies in a cell, whereas the mitochondrial genome may have 10,000 copies in the cell. And so most of what you're going to see is the real mitochondrial genome and not these annoying numites. But this integration is still happening. Studies went looking for polymorphic integrations and could find some new ones that are happening now. Like here, they found in one person 16,000 base pair recent integration into the intron of this gene. So this is happening. If it gets into the wrong place, you could imagine that being a cause of disruption of a gene. So this is an ongoing process. So let me step back and tell you how we got to this place. How did mitochondria evolve in the first place? So let's roll back evolutionary time. So all organisms on the planet that we know of have a common ancestor. And all organisms on the planet can be divided into 3 big domains with nothing in between. All organisms are either bacteria, archaea, or eukaryotes. Bacteria and archaea, to our eyes, look kind of similar. They're single cell, they're very small, they have very stereotyped shapes, they have no internal structure, the DNA is just loose floating in the cell. Eukaryotes, everything that you've ever seen in your life, except if you look at it under a microscope, is a eukaryote. The cells have enormous complexity and structure. They have mitochondria, they have ER, they have nucleus, all of these things. And they arose probably once, actually we know this, they only arose once, by the fusion of at least one archaea and at least one bacteria. So the nucleus and all of the genes involved in information processing derive from an archaeal ancestor. The genes, almost all the genes for metabolism come from bacteria. And there's a lot of theories about how this happened. And I love this topic. I could give a whole hour about the evolution of these early eukaryotes. But what we do know is that an aerobic bacteria, which is the precursor to the mitochondrion, is probably an endosymbiont and either a parasite or a commensal bacteria living within the cytoplasm of an archaeon. Now, we know it was an alpha proteobacteria because the sequence of the mitochondrial genome is most similar to that clade of bacteria compared to anything else. And they kept this double membrane, but very quickly in eukaryotic evolution, this bacteria that probably had 4,000 genes when it first started living in the host cell, it very rapidly lost almost all of its genes over to the nucleus. And why would this be so? Well, you can see this happening today. If you have a cell that gets a bacterial infection, those bacteria living within another cell start mutating and having reduced evolution very quickly. And the reason is energetic. So bacteria can divide so fast. E. coli can divide in 20 minutes. If there were endless resources, in 2 days, bacteria could have the mass of the planet Earth. That's how fast bacteria are dividing. So any small changes to the fitness will let the more fit bacteria take over among the other bacteria that it's competing against. So if you're the ancestor to the mitochondria, you've successfully invaded your host cell and you're dividing in that host environment. If you can lose genes, you will divide faster. So for example, bacteria have a cell wall. That takes about 200 genes to maintain a cell wall. If you don't need a cell wall anymore in the host environment because it provides you a lot of the stuff you need, like protection, if you can lose those 200 genes, not only is your genome smaller and you can divide faster, but you're not creating those 200 proteins. And each protein is, say, 200 amino acids. Every amino acid bond takes 5 ATPs to generate. So you have 200 proteins times 200 bonds times 5 ATPs times many proteins per transcript. That's a lot of energy that you don't need. And if you can get rid of those genes, you will divide faster. You will take over within the cell. So very rapidly, the genome from this ancestor went from a genome probably of something like 4,000 to something very, very, very small early on. And then probably some of these genes migrated to the nucleus, like the pneumites. That helped create a lot of the complexity of the proto-eukaryotic cell. These proto-eukaryotes evolved quickly and then radiated very quickly, and then there are all these different kinds of eukaryotes that you see today. All of them have the same internal structures, but have hugely diverged since then, and this happened over 1 billion years ago. Make sense? High level? Okay. So if we look at the remnants, this genome, the mitochondrial genome across different eukaryotes, they look very, very different. They go from 6 kb to almost 500 kb. But in terms of genic content, shown in purple, there's very little genic content. The genomes that have a lot of other mass, it's really just extra junk. Some have gotten introns and large non-coding regions. But there's very few number of genes in any organism that you see out there with mitochondria. So that supports the idea that they lost most of the genome very early before eukaryotes radiated. The most number of genes in a mitochondrial genome is 100. The fewest says 6. I know of one that has 3. And some organisms have actually lost the mitochondrial genome altogether. These are all organisms that are anaerobic and don't do oxphos anymore. And that's let them lose the mitochondrial genome altogether. Just this past year, the first eukaryote organism without any remnant of a mitochondria was found. It's a weird bug called Monocercomoides. It's a unicellular organism with 4 flagella. It's a beautiful organism, and it was found in the gut of the researcher's pet chinchilla. And this, we'll probably find some other ones, but it used to have, it derived from organisms that had a full functioning mitochondria, and once it became anaerobic, it lost the mitochondria. And in terms of the architecture, we're used to the human mitochondrial genome, this beautiful little loop. If you look across eukaryotic evolution, it's more like anything goes. There are ones that are linear, there are many linear ones, there are huge linear ones with telomeres, some have introns, some don't. So in 1 billion years of evolution, a lot of innovations have happened, and we have one of the more simple ones. So since we're partial to ourselves, I'll be talking more about the mitochondrial one in humans. All right, so for mitochondrial genomics, this has been revolutionized by deep sequencing, like everything else in genetics. And the mitochondrial has certain challenges that make it a little bit more of a pain. First of all, it's circular. So when you're mapping your reads, it doesn't map very well to the circle, and the coverage falls off at the ends. Now, the ends are the hypervariable region that's different among people and is doing less anyway, so it tends not to be a problem, but it is a little bit annoying. Another feature is that for germline genetics, you're used to thinking of 2 chromosomes per cell. Now you have to think of a system where there are thousands of mitochondrial genomes per cell. So you can find a variant not at 0% allelic fraction, 50% het, or 100%, but anywhere in that spectrum of heteroplasmy. So computationally, that is much more difficult to capture, and we now use tools that were designed for cancer, for looking for somatic alleles to try to find mutant alleles in the mitochondrial DNA. There is this huge annoyance of these pneumites, these nuclear sequences of mitochondrial origin. And this is just a headache for mapping. It doesn't have There are ways around it, but just to, I'll get to that later. But otherwise, it's pretty standard. So it's not too hard to call these variants. And I'm working with the DSDE platform here to come up with a pipeline that will be used around the world to call variants on the mitochondrial genome, just like to make it just as easy as your nuclear variants. The coverage in the whole genome sequence is usually excellent because there's so many copies of the mitochondria. For whole exome sequencing, it really depends on the platform. Older exome sequencing covered the mitochondria just fine. Then they tried to get rid of all the off-target reads, and they add blockers to the mitochondria, so you actually get very low coverage in the ICE genomes. So at the Broad, whole genome 20x nuclear DNA coverage gives you 2000x coverage of the mitochondrial genome. Whole exome sequencing, the ICE gives you an average of 30x, but it's so uneven that the median is only 9x. It's very hard to call variants with that low coverage. We helped the platform develop an extra method where they add back baits to the mitochondrial genome. So in your same tube, when you order exome sequencing, you can pay an extra $90 and get the mitochondria at 2000x coverage if you are interested in that for your disease. I'm working with DSD to make that pipeline. So now let's switch gears and talk about human mitochondrial disease genetics. So when we're talking about mitochondrial disease, we're talking about diseases that cause a defect in the OXPHOS system that generates approximately 90% of our cellular ATP. This is an umbrella term. It's a collection of many rare diseases, but together they have a combined prevalence of about 1 in 4,000 live births. Because mitochondria are in every organ system, you can have defects that present all over the body. Typically, you'll see defects in the brain, the muscle, often heart, but you can also get very specific defects in just one organ system, also caused by a lesion to OXPHOS. So because of the huge heterogeneity clinically, these can be very challenging to diagnose. Often the symptoms can be vague, and the diagnostic journey for these patients can be years of frustration and no one taking them seriously because they can't find a defect.
Sarah,
yeah.
What is the explanation for that? Is that the heteroplasmy and the tissue specificity, the variation? Because it seems to me, given the frequency of this, that the heart almost always ought to be involved because it's got so much mitochondria. Yet many of these don't have any heart disease. They have some other thing, like the retina, which has very little mitochondria. Yeah. So is that heteroplasmy as the explanation?
You read that in the textbook and it's wrong. So most OXPHOS diseases are not diseases in the mitochondrial genome at all. They're often recessive nuclear diseases, and they still have enormous tissue specificity. So we have a lot of hypotheses as to why this is. And so there can be the defect in the energy production system itself, but the body actually has a lot of ways of compensating. We're a complex system. There are a lot of ways of upping glycolysis to deal with the energy defect. But there are all these other downstream sequelae that I think are causing the tissue-specific pathology. And specifically for the retina, there's this disease LON, Leber's hereditary optic neuropathy. This is a disease caused by a mitochondrial mutation that is very mild compared to other ones. You have healthy young men in their 20s who suddenly have painless blindness, first in one eye and then a couple months later in the other eye, and then they're blind for the rest of their life. And they have a mutation in the mitochondrial genome that's at 100% in every tissue in the body, and the only thing affected is the optic nerve. So that is one of just exquisite tissue specificity. We think it may have to do with oxidative damage because you have a lot of oxygen and stress response there, but we don't really know. So that's at the end of the spectrum of some of the least severe mitochondrial diseases. The most severe are a class of diseases called Lee disease, characterized by these very specific MRI findings. And these are typically infants who in the first months of life have enormous failure to thrive. They have an MRI, see these lesions, and it's often lethal by the age of 2. So very sick kids. Some of these kids are the most expensive people treated at MGH for the whole year, even though it can be a very terrible disease. So you have this range of this pediatric lethal to this kind of mild blindness. And that makes it very challenging to diagnose these. Doctors use clinical criteria, something like this. There are 3 different criteria. This is the Morava criteria. And literally you get points. All right, does the person have muscle weakness? You give a point. Did they have CNS presentations like migraines and seizures? You'll get a couple points. You add these up, and if you have enough points, you have possible, probable, or definite mitochondrial disease. So you can imagine a lot of these symptoms are pretty vague. Like there are a lot of causes of seizures that have nothing to do with the mitochondria. So we were hoping when we got into this field, all right, we can clean this up with genetics. We're going to solve everything. And I'm not quite as naive anymore, but this is how the clinical folks are approaching these diseases. And even when you have a diagnosis, that ends the diagnostic journey for these families. But we can't really offer much. There are no proven therapies. Patients have all these questions about what's the course going to be for me, what's my prognosis, and the answer to pretty much all of them is we don't know. And so it's very hard diseases to treat. Genetically, they're extremely heterogeneous. We call them oxphos disease, but it's an umbrella of many rare diseases. There are over 145 genes that cause oxphos disease. And this isn't a GWAS with 145 hits of small effect. These are 145 different monogenic diseases giving rise to the same biochemical defect in oxphos. There are over 900 monogenic alleles with high penetrance, and no gene accounts for more than 8% of the cases. So it's very genetically heterogeneous. The mitochondrial genome, as we talked about, has 13 proteins. It accounts for about 15 to 20% of pediatric patients, and these are maternally inherited. The mitochondrial defects account for a larger percent of adult onset cases. These tend to be the more mild of the phenotypes. The more severe are defects in the nuclear genes. So there are about 1,200 proteins. My graduate work was figuring out what they all are. This is a list called mitocarda. And defects in these genes account for about 85% of pediatric cases. And the disease genes found to date are mostly recessive. There are a handful of dominant ones that are found and a couple excellent. But kind of anything goes with mitochondrial genetics. So these two heterogeneities of clinical and genetic make these perfect for genome sequencing to revolutionize the diagnosis of these diseases. And importantly, in most cases, from the symptoms, you can't say, all right, I think this person has a mutation in RS2 and this one has a mutation in POLG. Mostly with oxphos disease, you can't tell which gene is mutated in any given person. So the history of genetics for these diseases started with the first mitochondrial disease being described in the 60s. Mitochondrial DNA was first sequenced in 1981. The first mutations were identified in 88. And since as more genes have been identified, there's been traditional molecular diagnosis to sequence one gene or a panel of genes. So how successful have these been? Well, by 2008, the most severe, these pediatric lethal, infantile lethal ones, we could, by gene sequencing, candidate gene sequencing, get about half of the cases. In situations where it was a lot more fuzzy whether they actually had a mitochondrial disease, these gene panels could identify the molecular basis of about 18%. So after we published all the nuclear genes that go to the mitochondria, we said, all right, let's sequence those in patients with mitochondrial disease. We're going to really replace this awful diagnostic criteria with a single blood test. So we came up with mitoexome sequencing, applied it to over 200 patients, and yes, we can improve the diagnosis of different cohorts, but it doesn't solve the problem immediately. But this was just sequencing the mitochondrial genome and nuclear genes that encode mitochondrial proteins. Now we can do whole exome sequencing, but you see about the same thing. For really bona fide, biochemically proven mitochondrial disease, you get about half of cases where you can give a clear diagnosis. If you just have suspected disease based on these clinical criteria, about 40%. So there's a long way to go here. The genetics sequencing is really, really helping. Now this is becoming a first-line diagnosis. Instead of going from doctor to doctor over 10 years to try to get an answer, now a single test can give an answer in half of the cases. So that is an enormous improvement, but it's not good enough. 45%, we even have no candidate to look for. So either these are not a primary oxphos disease, or like any other genetic disease, it may be more complicated things. Non-coding regions, structural regions, maybe there's multigenic, maybe it's interactions between the gene and environment. I think this is very standard to what you see with a lot of other genetic diseases. Yep.
Have people done GWAS in the other 50% to look for complex traits?
No.
Why not?
I think in these cases it's less likely to be many variants of small effect and more small variants of large effect, but just lots of different ones.
Given the tissue specificity, that's what makes me think there's got to be multiple loci involved that give you some tissue specificity. But you're still thinking it's going to be all in the mitochondrial?
In the mitochondrial proteome. Because we know genes that are ubiquitously expressed still have extremely specific phenotypes.
Right.
So it doesn't have to do with the tissue. Let's talk later, because I think these are interesting ideas. So the main challenge ahead for mitochondrial human disease genomics is, like in most diseases, how do you interpret the pathogenic coding and non-coding variants? So at the last 5 minutes, I want to turn to somatic mutations that underlie just normal tissues or cancer. So in this space, there have been 3 really interesting studies in the last couple years, one actually coming out of this MPG population. So the first study looked at somatic mutations in the mitochondrial genome in 5 different cancers. And by looking at tumor normal pairs and off-target reads from the whole exome sequencing, they showed that there was a very high enrichment of non-synonymous versus synonymous mutations, and they concluded there's likely positive selection for mitochondrial mutations in cancer. A second study looked not at cancer, but just took autopsies, looked at different tissues from autopsy patients, and looked for mutations in the mitochondrial genome from 12 tissues each from 150 people. And they also saw very strong enrichment of non-synonymous versus non-synonymous mutations, also claiming positive selection of mitochondrial mutations. And then the most beautiful paper came along. I love this paper, so I'm going to tell you a little bit about it. And this is a little bit of a cautionary tale to people like me who are computational biologists who think, oh, let's come and run these tests and come up with conclusions. So they analyzed the same idea. They looked at tumor normal pairs from the ICGC, 1,600 tumor normal pairs. And they also, just like these papers, saw a 5-fold enrichment of non-synonymous versus synonymous mutations. But they looked really carefully at this and said there's no evidence of positive selection. And instead, they uncovered a really beautiful mutational process specific to the mitochondrial genome. And once you take that into account, these signals that look like positive selection just melt away. So this is a cautionary tale to really, with our statistical tests, you have to understand all the assumptions that are going in. So tests for positive selection are often based on this very simple notion of DNDS, also called KAKS, which is how many protein-altering mutations are there compared to how many synonymous mutations. So if you change in the coding region a nucleotide that doesn't change the protein, those should probably be neutral. But if they're protein-altering, that may be a sign of either positive or negative selection. So the number of non-synonymous substitutions per non-synonymous site over synonymous substitutions per synonymous site is this DNDS. And if you have more protein-altering mutations than you would expect, over 1, that would imply positive or Darwinian selection. If you have fewer than you would expect, that would imply purifying selection, that you're getting rid of these. And if it's about 1, then it's probably neutral. So that's the idea behind all of these. Does that make sense to people? Yeah. Okay, so here's the beautiful study. They looked at 1,600 tumor normal pairs. They looked at all variants that are over 3% heteroplasmy, and they found lots of variants. And a third of the variants were at hotspots that you found independently mutated in multiple people. In general, any given person in this cancer study had 0 or 1 mutations per mitochondrial genome. So you don't have a ton of mutations, but if you look at enough people, you'll gather enough mutations. The older the patient was when they got cancer, the more mutations you see. This is very typical. And different kinds of cancers had different rates of mutation, also pretty standard. And when you look at where these are in the mitochondrial genome, they cluster in the hypervariable region, shown here. And not surprisingly, like most things, 91% are transitions, going from C to T, or from T to C, or G to A, or A to G. So that's going from purine to purine, or pyrimidine to pyrimidine, not as big a change. So this is very normal. But then they noticed this gigantic strand bias. So most of the mutations are C to T, or T to C. But of the C to Ts, 84% of them are on the heavy strand. Remember, the heavy strand is the one that has most of the coding genes. So in general, the heavy strand has fewer Cs, that's why it's the heavy strand, but more of them are mutated. So there's 12 times more mutations that are on this one strand compared to the other than you would expect by chance. And similarly, of the T to C mutations, they're mostly on the light strand. So they thought about this for a while, and they showed, really interestingly, that there's one part of the mitochondrial genome that is replicated in the opposite direction from the rest of it. And if you look at that part, you see the opposite trend. So they concluded that this is a replicative process. The mutational bias that they're seeing is caused by a problem when you're replicating the molecule. And this is different than any other mutational process that they've seen on any nuclear genome. So I think that this is nice using high throughput genetics to understand some biology. And they then asked a really neat question of, well, this is happening in somatic mutations in cancer. Could this have also shaped the evolution of the mitochondrial genome over evolutionary timescales? And they showed that yes, in fact, it does. If you look at the codon table, it has been shaped by this. So of all the C to Ts that you could have, most of them have already been mutated from C to T. And so you don't see those as often anymore in our human genome. And if you so the codon table already fixes this replicative bias. So new neutral mutations are more likely to be non-synonymous than synonymous. And if you take this into account, then this positive selection goes away. So out of the 800 missense mutations, it looks like 4 to 1, but in fact, it's not positive selection. And on the nonsense mutations, you see as many as you would expect by chance, but they have lower heteroplasmy. So this study concludes that they happen at the same rate, but then they're selected against. So I wanted to show you this this is about how evolution and somatic mutations of mitochondria and how we have to be careful when we're analyzing our data. So with that, let me conclude, and I'll think some of the stuff that I talked about from our lab was thanks to my mentor, Wamsi, and thank you for your attention.